Hello students, myself Saurabh Yashaswee, Assistant Professor at Department of Zoology, RD and DJ College, Munger University, Munger. So today we will be discussing about a new topic, that is the receptors. So we have looked into the synthesis of hormones, how hormones are synthesized, and so we need to know about that how these hormones which have been synthesized are being received by the cells. Either they are being received on the membrane, or they enter directly into the cell or the nucleus, and then they bind to the receptor. So if we want to know that where they bind to, then we must know about the receptors. So starting with the slide, that what are the types of receptors which are found in the cells. We have classified the receptors into two types. Here you can see the first is the nuclear receptor and the second is the membrane receptors. So as far as the nuclear receptor is concerned, the name itself makes it clear that these receptors are located intracellularly into the cells. Either they may be present in the cytoplasm or they are directly present in the nucleus. So these nuclear receptors are the intracellular receptors which are present within the cells. So second class is the membrane receptors. The name clearly indicates that the membrane receptors remains at the membrane, at the cell membrane, where they receive the signal. So the membrane receptors have been further classified or categorized into four types, which are enzyme linked receptors, it can be cytokine receptor, it can be G-coupled receptor, and it can be ligand gated ion channel. So here if we look into the membrane receptors, we have four different type of membrane receptors which are present on the cell membrane. So now take us the example of each receptor one by one. So if we look into the enzyme linked receptors, we have tyrosine kinase receptors, we have serine threonine kinase receptors, we have histidine kinase receptors, and we have guanylate cyclases. So we will be discussing more about the enzyme linked receptors and other receptors as we will move forward in the topic. So here we will be confining to the type of receptors and what are the examples that may be, or the types of further receptors present in the those classes. So if we look into the cytokine receptors, we have soluble tyrosine kinases. Now when we go for G protein coupled receptors, G protein coupled receptors are transmembrane receptors which have a serpentine like structure, and we will be discussing more about G protein coupled receptors in the coming classes. So the next receptor is the ligand gated ion channels. When we look into the examples, we have cysteine loop superfamily, we have P superfamily, then we have epithelial sodium channels. So sodium ion channels, you should be knowing about sodium potassium channels, and so it can be easily recognizable with this example, the ligand gated ion channel. So these are the type of receptors which are generally found as a nuclear receptor or the membrane receptor. So we have classified it into the receptors on the basis of the localization, but further if we look, these receptors are of their own kind, and based on the localization we generally discuss it as nuclear and we discuss it as membrane receptors. So now take us let's have a look that what actually nuclear receptors is, and we will be looking further into the structure of the nuclear receptors, how is structured, how a structure nuclear receptor have. So now if we look into the some of the details of the nuclear receptor, we will come to know that what nuclear receptor is. So these are the simplest type of the receptors, and they are ligand regulated transcription factors. So these basically acts as a ligand regulated transcription factors, which means these receptors are localized within the cell, either in the cytoplasm or the nucleus. When the hormone or the ligand binds to them, then they regulate the process of, they bind to the DNA, and they regulate the transcription. So that is why they are the ligand regulated transcription factors. So as I am telling about the nuclear receptor since from the beginning, that they are intracellular, that means their localization is within the cell, and the ligand for these nuclear receptors are generally lipid soluble membrane. So when we discussed about the lipid hormones, we have already talked about that the lipid hormones directly enters into the cell, or they enter into the cell through they are present within the cell, and they directly bind to the receptors inside the cells. So when we talk about the lipid hormone, they get into the cell, then they bind into the bind with the receptors either in the cytoplasm, or directly they enter into the nucleus along with the receptors and initiates the process of transcription. So this is how the lipid soluble hormones generally act via the nuclear receptors. We will be looking into the mechanism further in the next slides. So here we have, when we look into the families of to which the nuclear receptors the act, then we have the glucocorticoid, then we have thyroid, and then we have estrogen, and then we have the orphans. So basically when we talk about glucocorticoid, thyroid and estrogen, that means that if we are talking about glucocorticoid and estrogen, we are talking about the steroid hormones, and thyroid. So since when we have talked about the classification of the steroid hormones, we have mentioned what are the steroid hormones. We have initially studied about the hormones classification, and we have talked about these things. And when we talk about here the orphan receptors, then I must tell you that orphan receptors are the unique receptors of which the ligand is not known, that the ligands are not known that to which they bind. So orphan receptors are often asked in examination, either in net examination or in common examinations, that what the orphan receptors are and what are their unique features. So one of the unique features of orphan receptors is that this receptors ligands is not known. So moving to the next slide, let's have a look that how does the nuclear receptor look, what is its basic structure. So when we look into the basic structure of the nuclear receptor, then what we can see here, we are having the three structures of the nuclear receptors to which the hormones bind, and the first is the glucocorticoid nuclear receptor. So A is indicating that the glucocorticoid nuclear receptor structure, B is the nuclear receptor that is of which the ligand is the estrogen, and the third is the triiodothyronine. So what you will see here, that these receptors have got some distinctive domains. So these domains have been marked with yellow color. So what you can see that they are terminals. The first terminal is the amino terminal and the carboxy terminal, which is the structure of, when we discuss about the structure of protein, we describe it as first the carboxy terminal and the amino terminal. So towards the amino terminal is the AB region, and then we have a DNA binding C region, then we have a hinge region, that is the D region, and then we have a ligand binding E region, and so occasionally you find there are the F regions that is extended beyond the E region. So when you look at the structure of the receptors related to glucocorticoid, estrogen and triiodothyronine, what you see in common is that they have distinctive 4 to 5 domains. And here you can see that each domain, suppose if I am talking about AB domain, then you can see it is in glucocorticoid it is written as tau1, in estrogen it's written as TAF1. Similarly, when we look into triiodothyronine AB domain, it is TAD1. So even within the domains there are different regions which perform different function. So let us have a look at the functions of each domain that is present in the nuclear receptor of glucocorticoid or estrogen or triiodothyronine. So when we talk about the domains, let us have a look what does AB domain indicates about. So when we talk about the AB domain, here what we have, that the AB domain is the amino terminal domain, and it is the most variable part of the nuclear receptor, and it doesn't appear to have a stable structure until and unless it binds to the DNA. So final configuration of AB domain is achieved only when it interacts with the other part of the receptors or with the DNA or with the transcription coactivator. So it is most variable part of the AB domain. Now the major function of the AB region is the transcription activation. So when in exams it is directly asked in net examination, the functionality of the domain directly and its associated function. So when we talk about the AB region, the major function of the AB region is the transcription activation. And when we talk about the transcription activation, then we must have transcription activating domain, that is TAD1, which we have talked about in the previous slide, just previous slide. And then there may be, when we have the transcription activating domain, so when we talk about TADs, we must know that they have their own property. So when we talk about the TAD, their TAD region, the transcription activating domain, exhibit gene selectivity. What does it mean, that what is the gene selectivity? So when we talk about the gene selectivity reflects the fact that many induced genes have requirement for specific factors. So if you need to induce any gene, then there must be a specific factors which must bind to it. So the uniqueness, the gene selectivity is therefore TAD, that is the transcription activating domain. If a required cofactors binds to the DNA, then only the certain gene is induced and the transcription can activate. So if we have a constellation of transcription factor, means some more transcription factors adds and comes and binds to the TAD1, then it can activate different set of genes than those bound to TAD2. So here we have TAD1, that is a transcription activating domain, which shows gene selectivity as the factors that binds to that region induce a specific gene, and when there is a constellation of transcription factor which binds to TAD1, then different sets of gene can then be activated. So this is all about. So this is all about the transcription activating domain. So there are some other domains also, such as TAF1 and TAF2. So we have TAF1 and TAF2. One of them is present in the AB region, and we will looking into the TAD. There are some more important properties of TAD also, and when we will look into TAD, we will find that beyond gene selectivity, it has got some other properties also. So here, when we talk about TAD region, we have talked about the transcription activation domain, and we have talked about gene selectivity. Now here we are talking about TAF1. So when we talk about TAF1, we have seen in the previous slide, it is associated with estrogen nuclear receptors. So this TAF1 requires higher air concentration. We have TAF1 and TAF2 both. So TAF1 and TAF2 have both different functionality. So if we look into the estrogen part, we will see that if TF1 of ER is estradiol independent, while TAF2 is estradiol dependent. So if it requires higher concentration of estrogen hormone to get activated, to make the receptor activated. So TAF can have different functionality in respect to being hormone dependent or being hormone independent. So this is the part where the domain activity can be influenced by the receptor concentration, and it is specifically TF1 or TF2. So beyond this, if we talk about the domains of the nuclear receptor, AB domain of the nuclear receptor, it has called receptor synergism. So the next property, first property is gene selectivity, and the second part is the receptor synergism. So what does this synergism means? It refers to the phenomena where transcription activation of multiple HREs is greater than sum of the effect of individual HRE. That means if we have multiple hormone receptor, if we have multiple HRE, then if more than one HRE gets activated at a time, then the transcription factor is highly stimulated. So if many transcription factors bind to the different regions of the HRE, then we have got a synergism. We have got the phenomena where the transcription activity by multiple HREs gets activated, and the effect is much higher as respect to the transcription done by the activation of one HRE. So this is the receptor synergism, and when we look into receptor synergism, it has been seen in the TAD1 of the glucocorticoid, estrogen, progesterone receptors. So the mechanism of transcription activation is thought to involve the binding of TAD1 to the transcription factor through coactivators. So when we talk about that what is the process of transcription activation, so we have that it involves the binding of TAD1 to transcription factors through coactivators. That means that coactivators are also being involved in the binding process. So what does the binding states that if we look into the synergism, if the membrane receptor is bound near to the gene, the more likely necessary factors will be attracted and stably retained in the vicinity. So if we conclude, we would like to say that the transcription, we would say that if we like to conclude about the AB domain, then we can easily say that yes, that the AB domain is responsible for the transcription activation, and it has got transcription activating domains which shows gene selectivity, and is often responsible for HREs, that is hormone responsive elements, and then it acquires the final structure once it fires bond to the receptors or the DNA or the transcription coactivators. And the next important part is the receptor synergism, which we have seen in the next slide. So here it is all about the receptor synergism, in which we talked about that the transcription factors by multiple HRE has greater effect than the individual HRE, hormone responsive elements binding, and we have seen that the binding of TA to a transcription factor may be involving a coactivator, and if a receptor binds more nearer to the gene, it is more likely that the factors will be attracted and the stability will be retained. So that will help in transcription process. So when we look into the next part of we have looked into the AB domain. Now we will look into the C domain, and when we look into the C domain, then one can associate that the C domain consists of two zinc binding sites, and these two zinc binding sites are finger-like projections which loop out and when they wrap around the DNA. So when we look into the zinc binding sites, there are two pair of cysteines located at 10 to 15 amino acid apart, and these are important for the specific contact with the DNA bases. So what we have that when we look into the structure, when we look into the structure of this DNA binding domain of the C region, which is the DNA binding domain, the name clearly indicates that it will bind with the DNA. So this DNA binding region has two zinc binding sites, and they have two alpha helices. When we look into structure, they have two alpha helices, and one at the these two helices are separated by 10 to 15 amino acids, and these are finger-like projections which projects out and they wrap around the DNA. So when we look into the helices, we have amino terminal helices that lies in the major group and makes the sequence specific contact with the DNA base. So now along with this helices, there is a P box which is located at the amino terminus of the first helix and recognizes the HRE, hormone responsive element. So this region is responsible for sequence specific contact with the DNA base and recognizing the HRE. So beyond this, we have other structures also which helps in dimerization. So let's have a look on that. So when we look into the structure, the next structure, we'll find that beyond this P box we have DR box, which there are some of the other factors which helps in dimerization process, which is the direct repeat called as DR box or D box, C2 residue at the second finger, A box, and this binds to the minor group of ATH 5', to the HRE, and also improves the DNA binding by stabilizing the DNA binding domain. So A box here is responsible for stabilizing the DNA binding domain. So next is a T box, which is responsible for the dimerization, and then we have nuclear exit signal, and it is located between the two fingers and shift the receptors into the cytoplasm when it is unoccupied. So when we look into the C domain, basically this C domain is the DNA binding domain, which have cysteine loop, and then it has P box, which helps in recognition of hormones responsive element, and then it has A box, which helps in stabilizing this DNA binding domain, and other factors, other parts like D box, C2 residue, which helps in dimerization, then T box, that also helps in dimerization, and one of the most important part is the nuclear exit signal, and this nuclear exit signal is present between the two fingers, and this helps in shifting the receptors into the cytoplasm when it is unoccupied. So if there is no hormone binding, then these receptors are then being targeted again to the cytoplasm, and this is due to the presence of nuclear exit signals, which is located in the C domain. So this is all about the C domain. Now we will look into the D domain and E domain. So when we look into the D domain of this structure, we will find here that D region is located between the DNA binding and the ligand binding domain, and its major function is nuclear localization. So it has got nuclear localization signal. So if its function is nuclear localization, it must have a nuclear localization signal, and this is recognized by the nuclear transport system, right? So this is the hinge part of the DNA, and it helps in DNA binding and dimerization, and few transcription cofactors and depressors have been shown to bind to the hinge region, and one of the most important steroid receptor, that is a steroid responsible for binding of estrogen, has this high mobility group B protein, which increases DNA binding for this NR. So basically this hinge region, as the name indicates, has the nuclear localization signal, and they participate in DNA binding and dimerization, and as well we have seen that it has some transcription factors which can bind to a few repressor or coactivators, and it can increase the DNA binding affinity, as we have seen in the steroid receptor. One of the factor is the high mobility group B protein, which increases the DNA binding affinities of them. So these structures have their own specificity, they have their own functions, and as a whole they make up the nuclear receptors, and then allows the binding and mobilization towards the nucleus. So when we talk about the E region, E region is also known as ligand binding domain. So we have seen the DNA binding domain. Now here the E region is the ligand binding domain because of the hormone binding. So now this region, in the E region, it is the ligand that binds to the receptor. So this E region of the receptor is responsible for ligand binding, and this ligand can be hormone. So this ligand binding is three-tiered, a helical sandwich layer. Each layer of helices is approximately perpendicular to the adjacent layer, and it has transcription activation domain 2. So when we are talking about the transcription activation domain 2 here, so we have earlier talked about the TAD1, and that was in the AB region. So TAD, that is the transcription activation domain, there are two in numbers. The TAD1, it is present in the AB region, and TAD2, it is present in the E region of the nuclear receptor. So these are the this is the important function of the E region of the nuclear receptor, that it binds to the ligand, and when it binds to the ligand, it initiate other functions like transcriptional activation, dimerization, and heat shock binding, protein binding. And when we talk about the F region, then we have it is an extended region after E region, and it is the carboxy. It is the region present at the carboxy terminal to the ligand binding site, and this region in estrogen receptor, nuclear receptor, has been found to bind some of the co-activators. So we have talked about the nuclear receptor, we have talked about the structure. The structure of nuclear receptor consists of A, B, C, D and E and sometimes F regions. We have talked about the AB regions, that it has got transcription activation domain 1, it has got TF1, it is responsible for transcription activation. Then the receptor region has gene selectivity as well as it has got the synergism. So this is confined to the AB region. When we have talked about the C region, we have talked about the DNA binding region. So it is the region that binds to the DNA selectively in a sequenced way, and then it binds to the HRE, hormone responsive elements. And then we have talked about the cysteine loop that is present there, and then we talked about the D region. Then we have talked about the E region, which is responsible for the ligand binding, and then we talked about the F region. So here the receptor regions are important for their own, because they have their own functions, they have their own properties, and these properties are oftenly asked in the NET examination. So we have talked about the receptors, and in the next what we have so in the next part we will talk about how these receptors gets activated, how these receptors will be responsible for the function of the steroid hormones, and how this steroid hormones get how these steroid hormones binds to the DNA and then stimulates the transcription factors. So that is for the next topic. Till then, stay at home, stay safe.